Okay, I almost got finished with chapter 8 on Tuesday. It was longer than I thought it was. And so I got almost to the end. And chapter 8 is all about chromosomal level mutations, which are big changes in chromosome structure, and variations in chromosome number, and perhaps variation in gene number. And let's see, I got all the way down to it was longer than I thought. Okay. There is an event that happens during cell division. And during correct cell division, the chromosomes are going to have to segregate into the two daughter cells, and they're going to have to segregate in the appropriate numbers and the proper, um, the appropriate numbers of each chromosome. And so a failure of chromosomes to separate is called non-disjunction. And there are some ways that chromosome number can vary. And you can have non-disjunction during meiosis, which is fairly common. And you can have non-disjunction during mitosis, which is fairly uncommon because non-disjunction during mitosis tends to make both daughter cells have abnormal numbers of chromosomes, and that tends to be lethal. And you can make what is called alloploidy, which is a cross between two different species. Now, non-disjunction just means failure of chromosomes to properly segregate. And during meiosis, non-disjunction can make cells that have too many or too few chromosomes. And non-disjunction can happen in meiosis 1 or in meiosis 2. If it happens during meiosis 1, in anaphase 1, every gamete that is produced will be abnormal. Half will have, and you assume it happens once. It's easiest to imagine this error happened one chromosome only. And so every gamete will be abnormal. Half of them will have one too many chromosomes, and half will have one too few. If it happens during anaphase 2, half the chromosomes will be completely normal, and half will be abnormal. One will have one too many chromosomes, and one will have one too few. Now, usually non-disjunction is partial. It typically happens to one chromosome, maybe two. In certain instances, you can have what is called complete non-disjunction. That means it results in one of those daughter cells having all the chromosomes and one with none. The one without chromosomes dies. You have no DNA, that's it. But you can have, if complete non-disjunction happens in meiosis, you can make a diploid gamete that you can fertilize with a typical haploid gamete, and it makes a triploid individual. Now, in mitotic non-disjunction, it occurs after fertilization, and it's usually only going to be a subset of cells that are affected. And if it happens during mitosis, the chromosomes separate improperly, and it can lead to trisomic or monosomic daughter cells. Alternatively, what can happen is you can lose a single chromosome. One of the sister chromatids does not migrate.
Quick question.
Yep, yep.
So for exam 2, it's going to be due at noon again, correct?
I'm going to decide when to post it,
Okay, that's
and I
fine.
will make an announcement, so it won't come as a surprise.
Okay, thank you.
I kind of like this noon thing better than the midnight thing.
Yeah.
Anyway, chromosome loss can also result in cells that can be normal or monosomal. And so in mitotic non-disjunction, you have the sister chromatids that fail to separate. And so one cell is trisomic and one cell is monosomic. And that is usually a lethality. And in chromosome loss, the microtubules that move the chromosomes around during cell division sometimes disassociate. And if that microtubule breaks off, that chromosome that had the microtubule break off is just adrift. And usually, what will happen is it could be left outside the nucleus during that last part of mitosis. And then the cell will read that as erroneous DNA, and it typically destroys it. And then you have what is called mosaicism. And a mosaic region just means that there are little patches of tissue that are going to be phenotypically, and in this case, they're going to be genotypically different from each other. And this happens all of the time. And the little mosaic areas are usually too small to really be observed. They happen fairly late. And so they happen after there's quite a bit of cells in the zygote. If, however, you have this non disjunction happen in the very first mitotic division after fertilization, it means half the cells of that organism's body will be one type of genotype and half the cells will be another. Now, the prefix auto is when you take, for example, in gamete formation, you have complete non disjunction. And so you have a diploid species that then makes diploid gametes. But you use those diploid gametes to fertilize other diploid gametes from the same species. And so you make an autotetraploid. And auto just means self. And now, if you do something called alloploidy, you can result in crossing two species. And in this case, in this picture, the individual that would be produced here would be an allodiploid, because the prefix allo means different, but you still have two of the copy number of the chromosomes. And you can have what they call allopolyploidy. And in this case, you have species one, which is a diploid. You've undergone something to make it do complete non-disjunction. And you have species two, which is a diploid, and it undergoes complete non-disjunction. And so you make diploid gametes. And you use those diploid gametes to fertilize each other. And you make an allotetraploid. This is actually quite common in plants. It's much, much more rare in the animal kingdom. Now, we can make things polyploid, and we have done so, especially in breeding crop plants. And you can do this in a variety of ways. Suddenly change the temperature during gamete formation or use of drugs during gamete formation. And a very old, well understood, well used one is the drug colchicine. And colchicine binds to tubulin, which is something that is found in the spindle apparatus. and it causes those microtubules to decay. And so you typically get complete non-disjunction. And so you have a normal diploid plant. Part of it with colchicine. And so now you have a little patch where that plant is tetraploid. And in plants, you can usually tell because the tetraploid plant will usually be, well, I don't want to really say healthier, but you can measure the rate of growth or the size of the plant or the amount of flower set or something. And usually the polyploid plant will be bigger or more robust or have bigger seed sets or something. And in a lot of plants, what you can do is you take a cutting, and if you treat it right, you put it under correct conditions, it can reroot itself into a substrate, and it can resume growing into a full tetraploid individual. Now, chapter 9, which I was just taking a look at, in, I believe it was chapter 7, it talked about linkage and mapping and whatnot in a eukaryote. And in chapter 9, it goes through linkage and mapping in a prokaryote. And it usually is going to refer to just plain old everyday bacteria, because bacteria are the easiest to work with and the easiest to understand. And scientists have known, biologists have known for about, at least 80 years or so, that bacteria can quite readily exchange DNA with their environment and with other bacteria. And there's some real world applications for this. Bacteria and viruses are the primary disease causing agents in, well, lots of things, humans included. Bacteria often possess differences in their genotypes that affects the differences in the phenotypes that they express. And for example, bacteria can be sensitive or resistant to antibiotics. Now, bacteria are almost always haploid. And working with a haploid organism is, in a lot of ways, easier because you only have one copy of every allele. And so that any kind of recessive mutation can't hide in a, like it can in a diploid. It can hide in the heterozygote. Bacteria typically, we say, produce asexually. They reproduce clonally. And so you have to be a little bit different in how you work with bacteria. Now, we do know that bacteria can transfer DNA from one bacterium to another, and it can be of the same species, and it can be of different species. And that can be useful for locating a gene on a bacterial chromosome. There are 3 basic mechanisms of genetic transfer found in the bacterial domain. And there's what we call conjugation, which is direct transfer of genetic material from one cell to another. There's transduction. Viruses can transfer DNA between 2 types of bacteria. And there is transformation. Some bacteria can uptake DNA from their environment. So there's conjugation. Conjugation requires direct contact between two bacteria. There's transduction. A lot of times, viruses, when they infect cells, a virus will always incorporate its genetic material into its host cell's genome. And sometimes, when that viral genome extracts itself from its host cell, it pulls some of the host's DNA with it. And when it infects the next cell, some of that DNA is injected into the host. And in transformation, how it was discovered, you have a bacterial cell of one type. And especially, this is true if you kill it. If you kill things, one of the things that happens is their cells burst open. And in the case of bacteria, their DNA goes out into the environment. And sometimes recipient cells can absorb this DNA and incorporate it into their genome. Now, conjugation, research group, Lederberg, Tatum, and Davis. Conjugation is direct transfer of genetic material via physical contact. And there is a type of plasmid. And a plasmid is a tiny little piece of extra nuclear DNA that you find in usually bacteria. And there's all kinds of different plasmids. And there's somewhere there's a list of them. Anyway, some plasmids allow bacteria to transfer DNA between bacteria to bacteria. Some plasmids make the bacteria resistant to certain antibiotics and all kinds of things. Plasmids usually have genes on them that provide some use, because if a bacteria has a plasmid, it means it's slower to replicate all of its DNA at every time it divides. And so if that plasmid does not convey some, you know, benefit to the bacteria, it will very gradually be replaced by bacteria that don't have plasmids. And so this research group were studying E. coli and had differences in the types of nutrition they needed to grow. And they had something called an auxotroph, which can't make a needed nutrient it needs to grow. And they had a prototroph, which is something that can grow on what they call minimal media. It can make all of the nutrients it needs from a very basic set of conditions. And so Lederberg and Tatum were working with two auxotrophic strains, and one was bio minus, met minus, but then threonine, leucine, and thiamine plus. So it could not make biotin or methionine. And the other was the opposite. It could make biotin, methionine, but it couldn't make threonine, leucine, or thiamine. But you need all five of these things to be plus if the bacteria is going to grow on minimal media. And so they had a bunch of strains. They had diluted these mixtures all to the right concentration. And in one tube, they had one auxotroph. And in one tube, they had the other. And in this center tube, they had mixed together auxotroph strain number 1 and auxotroph strain number 2. And they would let that sit for a while. And they would take samples from all three tubes and plate it on minimal media. And bacteria from this tube couldn't grow because they couldn't make these methionine or biotin. The bacteria from this tube couldn't grow because they couldn't make these three things. But when they played out the mixture, they got some colonies. And so something had happened when these bacteria were mixed. And so one of two things could have happened. It was probably the first one. This is simpler. And so it was probably this that happened. And so either met minus bio minus and then threonine leucine thymine plus obtained the ability to make biotin and methionine, or the other strain gained the ability to make threonine, leucine, and thymine. And that simple experiment couldn't tell what had happened. And then a further experiment showed that this bacterial conjugation could not occur if you kept the bacteria in question from coming into physical contact with each other. And so Davis had these two auxotrophic strains in a U tube, but it was the two halves were separated by a filter. And the pores of this filter didn't allow bacterial cells to go through. It would allow the liquid, and it would allow anything smaller than a bacterium that was in the liquid to go through. And basically, under a variety of pressure changes, this liquid was sloshed back and forth. And when all of these, things after a while were plated, nothing grew on minimal media. And the conclusion was that since they did not come into physical contact, there could be no exchange of DNA. Now, conjugation is direct transfer of DNA from one cell to another. A lot of bacterial species can conjugate, but not all. And the ones that are capable of this have a plasmid called an F factor. And F just means a fertility factor. If a bacterial cell has the F factor, it is designated F plus. If it doesn't have it, it's designated F minus. And the F factor has genes that allow for the transfer of DNA. And so what happens? An F plus cell can conjugate with an F minus cell, and that will result in the conversion of that F minus cell into an F plus cell. I added this little thing on it. This is straight from the textbook. Now, this is the F factor. And it says there's a couple of dozen genes on here that are necessary for conjugation. And they are all given the names TRA, some capital letter, TRB, some capital letter. And a few of those genes have been identified in this image. I'm sure the other ones have been identified as to what they do. They're just not named in this image. Now, conjugation is formed or is started with the formation of what they call a sex pilus. And these pili are only made by F plus strains. And so there is a pilus between the two bacteria. And so the big events, sex pills contacts the F minus strain. There is a protein complex called the relaxosome that binds at the origin of transfer in the F factor DNA and cuts one strand. The relaxosome separates the strands. The relaxase is part of the relaxosome, stays bound to one of those single strands. That complex of DNA relaxase is pumped out of the donor cell into the recipient cell. And after the transfer is complete, the relaxase reknits that plasmid into a circle. So wherever the origin of transfer is, you cut it there, the sex pulse is made, you begin to unwind the plasmid and transfer it into the recipient cell. And then the DNA repair mechanisms basically rebuild the double-stranded plasmid. And you've taken this F minus cell and converted it into an F plus cell. Now here's the plasmid. I knew this was here. A plasmid is a piece of DNA that exists outside of the chromosome, and it's usually a circle. All the ones I've ever worked with are circular. And it says they occur in bacteria and yeast. All of the ones I've ever worked with occur in bacteria. And they're not that big. Few thousand to 500,000 base pairs. All of the ones I've ever worked with were like maybe 15, 20,000 base pairs. And they range in size and the number of genes they have. Maybe a gene, maybe a couple of genes, maybe 100 genes. And there's a special type of plasmid. It's called an episode. And this plasmid can integrate into the bacterial chromosome. And an example of an episome is an F factor. And so there are types of plasmids: fertility plasmids, resistance factors. They supply resistance to antibiotics. There are plasmids that contain genes that allow you to make enzymes for metabolizing unusual things. Some plasmids allow certain bacteria to kill other bacteria. And some plasmids can convert a non-virulent bacterium into a virulent bacterium, non-pathogenic to pathogenic. Now, you can use this episome effect to do genetic mapping. And it was found in the 1950s. You just stumbled across this. There was a type of E. coli that became known as the HFR strain, high frequency of recombination. And in this case, it turns out the F factor is integrated into the bacterial chromosome. And so you can make HFR strains from F plus strains. And it turns out there are many locations on the bacterial chromosome where this integration can happen. And the F factor can excise itself from the chromosome. It can cut itself out and drag with it some of the genes that were once in the bacterial chromosome. And those types of F factors are called F prime factors. And it turns out that F prime factors can be transferred through conjugation, and it can carry new genes along with it and alter the genotype of the recipient cell. And so here you have the F plus cell, the F factor is separate. Here you have the HFR cell. The F factor has been stuck in there. And during the transfer, here is your place where the origin starts. And you have a couple of genes on this chromosome that you can mark. And it starts to transfer. And you can actually time the amount of minutes it takes to transfer gene 1 or gene 2 or gene 3 or so on and so forth. And the longer it takes to transfer these genes simply means it's further away from the origin of transfer. And so in an HFR and an F minus strain, one of the DNA strands in the HFR strain is going to be nicked, which determines the starting point of the transfer. That strand of DNA of the HFR begins to enter the F minus cell. Because of the typical size of bacterial chromosomes, it has been determined that it would take a couple of hours to transfer the entire chromosome. A couple of hours is an eternity of time to a bacterium. And so usually this conjugation is interrupted before the 2 hours is complete. And so only some of it happens. In the recipient cell, the chromosomal region that was conjugated can recombine with a region of the recipient's chromosome, and therefore you can introduce new alleles. And so there's your HFR cell, your F minus cell. You begin to feed in the chromosome. And if the time has been very short, basically only one of these alleles, in this case lac plus, gets transferred. And at the other site, it's still pro minus. But if you let it go a long time, both of these genes get transferred into the recipient cell. And so mapping in E. coli is done by something called interrupted mating. It was developed in the 1950s. And it just means that the longer it takes to transfer the genes in various parts of the chromosome gives you an indication as to where on that chromosome those genes are. And so the idea was if you allow bacteria to conjugate, but you interrupt this mating at different times, you could begin to construct a map of genes on the E. coli chromosome. And so, for example, you have an HFR strain. And the HFR strain was threonine plus, leucine plus. It was sensitive to azide. It was sensitive to infection by a virus. It was lac plus. It was galactose plus. But it was sensitive to streptomycin. And the recipient strain was the exact opposite at every location. And it was already known threonine and leucine, threonine and leucine genes were first. And it was already known that they got transferred within the first few minutes. And so the goal was to figure out the order of the azide, the T1 susceptibility, lactose, and galactose positivity were transferred. Streptomycin was not examined. It was used to kill all the bacteria after the experiment. And so basically, mix the two types of bacteria together. You allowed them the conjugation to happen. But every now and then you would take your mixture of bacteria and put them in a blender, turn on the blender, and that the idea was that would separate the conjugation, it would separate the two cells, and conjugation would stop. And then you would plate the cells on medium, and basically, time after time after time, you would see what kind of media they could grow on and what they couldn't. And if you repeated that enough, you would get something that looks like this. Interrupted the mating in 5 minutes, you saw basically nothing happen. Conclusion was no genes had been transferred after 5 minutes. Let it go 10 minutes. Cells, after 10 minutes, threonine and leucine had been transferred. And there were a couple, increasingly rare did you see any other genes get transferred. And then you would let it go for 15 minutes. And well, after 15 minutes, threonine and leucine had all been transferred. And in about 70% or so of the experimental values, you saw a transfer of this azide. And in about a third, you saw a transfer of the T1 susceptibility, but no lactose or galactose. And so they began to put these things in order. And then let it go for 20 minutes. And after 20 minutes, you see all of the cells with this threonine leucine. You get a greater number of this azide, a greater number of the T1 susceptibility. And finally, you start to see the lactose positive. Let it go 25 minutes. All of the recipient cells are now threonine leucine positive. Most of them are azide susceptible. Most of them have become susceptible to T1 infection. A pretty good percent are now lactose positive, and you're starting to see the presence of the galactose. And you kept on going 40 minutes, and so on and so forth. And you would, as you went longer and longer and longer, get more and more and more of the genes transferred to the recipient. And so, period. You can make a map just based on how long it takes for certain genes to get transferred into a recipient bacteria. And experiments like these have been used to map 1,000 or so genes on the E. coli chromosome. And we say that the E. coli map is 100 minutes long. It takes about 100 minutes to transfer the complete chromosome in an HFR mating experiment. And so when you map genes in this E. coli, you put the genes in minutes. How long does it take to transfer these genes without interruption until you get all the way around the chromosome? And you can calculate the distance between genes. And again, you measure them in minutes. And you can get an approximate time of how long it takes to get transferred by just back tracing it to, the one previous. And so in the case of LAC Z and Gal E, we say they are separated by about 9 minutes on the E. coli chromosome. And notice we don't use like centimorgans or anything like that. We just use minutes. Now, this is transduction. Viruses can transfer DNA from one host to another. And it doesn't have to be bacteria to bacteria. It doesn't even have to be the same species of bacteria to the same species of bacteria. It can be virtually anything to anything as long as the virus in question can infect both things. But we're going to simplify. Transduction is a transfer of DNA from one bacterium to another via a phage. And a bacteriophage is a virus that is specifically able to infect bacteria. It's got a very simple construction. Piece of genetic material surrounded by a protein coat. And these bacteriophages have really boring names. For example, there's P22, which infects Salmonella. And there's P1, which infects E. coli. And there's hundreds, if not thousands, of more of them. And the way a virus generally works, the phage infects a cell by injecting its genetic material into that cell. In the case of a phage infecting a bacterium, the bacterial chromosomal DNA is digested into little bits. New phage proteins are assembled, and you package that DNA into those new little protein coats. And sometimes mistakes are made. And so you get pieces of bacterial DNA along with the phage genome packaged into the phage protein, which then can now lyse and rupture, release the phages. The phages can go infect another cell, and that little piece of chromosome that might have in error been packaged in with a new phage can go in to the second bacterium. Now, this last one is bacterial transformation. It was discovered kind of by accident back in the 1920s. And if you took biology and one for majors, you got through the outlines of this experiment. And in this experiment, Griffin was working with the smooth strain and the rough strain, and the smooth strain could cause pneumonia, and the rough strain couldn't. And he found out that if you mixed living R strain with heat killed S strain and injected a population of mice with the results, the mice could get pneumonia. And when he did a biopsy, he would find living S strain bacteria in the mouse's tissues. That was an example of natural transformation. Those cells were just what they call competent. You didn't have to do anything fancy to allow them to uptake DNA. Some cells you have to use special techniques to make them uptake DNA. Anyway, natural transformation occurs when bacteria are quote unquote competent. And these bacteria carry genes that encode proteins that are called competence factors. And those proteins allow the bacteria to bind two pieces of DNA, uptake them into the cell, and incorporate them into the bacterial chromosome. And so, in transformation, well, a big piece of DNA binds to the surface of the cell. Enzymes cut the DNA up into little pieces. One strand of that double stranded molecule is broken apart. The other strand enters the cell. That other strand must be incorporated into the chromosome to be stable. The ideas about how this happens kind of hand waving. We think something called homologous recombination is involved. This results in DNA that has mismatches. DNA repair enzymes detect those mismatches, and they fix those mismatches. And sometimes they will transform the recipient to something new. And sometimes those repair enzymes will simply get rid of the mismatches and not transform the recipient into a new genotype. But anyway, there's a receptor on the bacterial cell. Big piece of DNA binds to it, chops it into bits. One strand is brought in. Lysine minus and lysine plus, same gene, two different alleles. And so they're very, very similar in sequence. But that allows these to homologously align, and you can basically find these little mismatches. And then using a variety of repair mechanisms, switch this lysine minus gene, which was native to the recipient, to a lysine plus gene, which is what got transformed into the cell. Sometimes DNA that enters a cell through transformation is not homologous to anything in the recipient chromosome. And so it might be incorporated randomly into the chromosome. And this is called non homologous recombination. And don't ask me for details about this because I don't know them. Now, this all has medical relevance. Bacteria, especially, are very likely to do something called horizontal gene transfer. We generally thought of gene transfer as between, for example, one generation to the next within the same species, parents to offspring, and then to offspring, and then to offspring, and so on and so forth. And that was called vertical gene transfer. And now we know that horizontal gene transfer is possible. Transfer of genes from one organism to another, maybe from one species to another, maybe from one kingdom to another, maybe from one domain to another. Common examples in bacteria are conjugation, transduction, and transformation. And we now know, I want to say now and know, we now suspect that horizontal gene transfer is much more common than was previously thought. Now, the medical importance of this. There is an increasing potential problem of antibiotic resistance in bacteria. And there are some bacteria that are resistant to virtually every antibiotic we know of. And some of these bacteria cause human illnesses. And for example, there's a bacterium, Staphylococcus aureus. And these happen to be resistant to methicillins and penicillins. And there's an example, MRSA, methicillin resistant staphylococcus. And the thing about staph is everywhere. A large percentage of the human population has staphylococcus growing on them, especially on the surfaces of their body. And they usually cause no problem at all whatsoever. However, these resistant staph can cause skin infections that are very hard to treat. And they can penetrate deeper into deeper tissues and damage deeper tissues. They can, in serious conditions, cause the death of the patient. But Staphylococcus is basically an innocuous bacterium. Most people have it living all over themselves. And it usually does nothing at all whatsoever. However, if it acquires this resistance, it can cause serious diseases because they start off as looking at like something innocuous. And then they can progress to something that's very, very, very serious. That is the end of chapter 9. Earlier today on Connect, I posted chapter 8. I will either do chapter 9 before I leave today, or I'll do it tomorrow. And so, therefore, I expect exam 2 to be the first half of next week. If I need to, I could extend it to the later half of next week. And I think I'll still have plenty of time to finish all the material in this course before the end of the school year. And so, any questions, best way to still get a hold of me is email. I told my class I had this morning, once upon a time, I got BRCC email sent to my phone. And for some reason that has expired. And I have decided that I kind of like not having BRCC email forwarded to my phone. And so I haven't tried to fix it in the last couple of years. But chapter 8 is on Connect. Chapter 9 should be there soon.